All right, 115. Um, questions on chapter, what, 3? All right. Started chapter 3 this week. Should be finishing that up this week. So the floor is open for questions related to lecture, lab stuff. Oh, quite a bit of stuff in chapter 3 there. Can you hear me? Okay.
Yeah.
Great. Have you looked at chapter 3 yet? I hope so. Anything you want to go over?
I listened to your lectures on chapter 3, but I was just wondering if you could just briefly review the different active transport. I just feel like I'm having a hard time differentiating between the like exocytosis, transcytosis, endocytosis, all of those.
Okay, great. Yep. I'm pulling up the PowerPoint as we speak. Great question. So let me share my screen. Do you see the PowerPoint? Yes, okay. All right, so let's see. Active transport. All right, so when we talk about active transport, the term active implies the need for energy. The cell has to expend what we call ATP, which is the primary energy currency of the cell, in order to move a substance basically against a concentration gradient. In other words, rather than going from high to low, like we do with diffusion, for example, which is passive, we're moving across that gradient, if you will. We're going against it. The metaphor that I often like to give is if you think of a salmon swimming upstream, having to jump some significant waterfalls sometimes to get back to the stream that it was hatched in 10 years ago, right? That fish has to swim against the current, doesn't it? And when it does that, it has to expend a lot of energy. You've all seen pictures of that, right? Movie clips or whatever in nature shows. So this is the process of moving against that concentration gradient, and the cell has to expend energy to do that. Okay, and as you will be talking about when you get into the nervous system, the way that certain neurons function employs this active transport process. In fact, I remember reading somewhere, maybe I stated this statistic somewhere in the PowerPoint, something like, I think it's 40% of a cell's energy usage goes into this very process of active transport. So it's a very important process that neurons, nerve cells, make use of, but other cells as well. And in order to move that substance against that concentration gradient, we have to employ the use of special carrier proteins. We call them transport proteins, carrier proteins, same thing. And this is a slide that sort of depicts that process. Here is our carrier or transport protein in purple. Here's our phospholipid bilayer, which we know is an important constituent of all cell membranes. And the little purple circle there, whatever you want to call it, the particle they're defining it as, is being moved from a lower concentration toward a higher concentration of that particle. And so what really ends up happening, and this is getting into more detail than you probably need to know, but the particle actually bonds to a specific receptor site here on the carrier protein, and then ATP comes on board. It's degraded, and that changes the shape of the protein, which in turn kicks the particle out or into the cell, whichever has the higher concentration. Okay, it doesn't define inside or outside of cell here. It just says lower concentration on this side of the membrane, higher concentration on this side. It could be this is the inside, that's the outside, or vice versa. But the point being, you're moving against the gradient, moving toward a higher concentration from a lower. That takes energy, and that energy is supplied by the cell as it degrades ATP. And that degradation of the ATP is what changes the shape of the protein. And that shape change is what kicks the particle out. Now, as soon as that particle gets kicked out, the protein goes back to its original conformation and can accommodate another particle, if you will, if there's energy to be broken down, and then we continue to pump the substance toward that higher concentration. And this YouTube link, I haven't watched it probably in about 3 or 4 months, but I'm sure it takes you through that very process. So I'm hoping you're watching these. You should be watching those embedded hyperlinks. Now, we also talk about specific movements of substances. In this case, we're going to be describing movements of materials into cells. So this is an active process. By that, I mean requires energy. And we described in the chapter 3 different mechanisms. The first of which is called cell drinking, or pinocytosis, some people pronounce it. So here, of course, is our cell with a nice nucleus and outer cell membrane. And so in this particular instance, we are bringing fluid from the outside of the cell into the cell via the formation of what's called a vesicle. So here's our vesicle. It really is made up of cell membrane, because that's where it came from, right? You can see the cell membrane invaginating here. And in the center of that vesicle, the cargo, if you will, of that vesicle is this outside fluid that the cell somehow wants to make use of in some manner. That requires energy for a cell to pinch off its cell membrane and make the vesicle. Phagocytosis literally means cell eating. So we're now engulfing some sort of a particle. For example, this could be a white blood cell. This could be a bacterium. Or this could be some sort of a food particle, some nutrients that the cell would like to degrade, for example. So in the same way that we formed a vesicle, the cell membrane invaginates. There's our vesicle. Only in this case, we have some outside particle that is the cargo. It's not liquid like it was earlier in pinocytosis. This is the introduction, if you will, of some sort of a, they call it a solid particle, a non-liquid. Phagocytosis. Most of the time when you see that word, it infers the immune system or white blood cells engulfing some pathogen. It doesn't always have to be that, but that's typically when you see that word, it's used in relation to some discussion of neutralizing a bacterium, let's say, or something like that. And then the last example of endocytosis is a very special case, endocytosis, called receptor mediated. And here we're looking at the presence of specific receptor sites, which are proteins, on the surface of a cell membrane. So here's the inside of the cell in orange, yellow. Here's the outside of the cell. And out in that extracellular fluid, we have various molecules. And we'll depict them as different colored, different shaped structures, because we know that molecules indeed do have their own unique three-dimensional conformation or shape. They're not really blue and red and yellow, okay? We're just making those colors up. But every molecule has a particular chemistry. Every molecule has a particular conformation. Case in point, when you think of DNA, what do you think of in your mind? What kind of shape? What's DNA's shape? Double helix, right? So just, think of DNA. But even a protein or a lipid or a carbohydrate, all molecules have shapes. This piece of fossilized wood has a particular shape or conformation. Everything has a conformation, except for maybe a water. Well, water molecules, I guess it's a molecule that has three-dimensionality too. So anyway, here we've got those receptor sites that will only accommodate specific shaped molecules. In other words, will the yellow or blue guys fit into the receptor sites? No, because their shape doesn't conform. The pink shaped or colored particles, however, do have a proper conformation such that they fit into those receptor proteins, as we have in the second box. Then what happens to the cell membrane? Well, same old, vesicle forms, right? Cell membrane invaginates. Here's our vesicle, only here the vesicle is carrying a special predetermined cargo as dictated by the shape of the receptors. Okay, that's the takeaway message. The receptors determine what's brought in. If you don't fit into the receptors, you won't be brought in this particular example. The blue and the yellow guys stay outside. Only the pink guys allowed in. This too takes energy. Whenever you pinch inward, whenever you pinch your cell membrane inward and make a vesicle, that takes energy. Okay, question.
And this is probably like more advanced than we need to go, but how does the cell use like whatever's in the vesicle after it's inside? Like how does it get outside of the vesicle to serve the cell?
Yeah, okay, that's a reasonable question. I'm not sure that there's going to be a figure in this book that shows this. Let me see. Oh, actually, this kind of does show it. Okay, so let's talk about another mechanism that will tie into your question. So it's really an excellent question to ask, because I think I can answer it here. And also talk about exocytosis. Now, remember, endocytosis was bringing something in. Exit, think of exit, we're leaving. So here we're talking about the release of something from the cell. Okay. This is also an energy expenditure process. It's not free to the cell. It does require some energy usage. So let's pretend here's our recently phagocytized particle that we just brought in via endocytosis. Okay, there's our food, if you will, quote unquote, that the cell somehow is going to make use of. So your question was, how does the cell use that recently phagocytized food? Here's how it can do that. It also has membranous organelles called lysosomes. You should have read about those earlier in the chapter. These are organelles that have powerful hydrolytic enzymes in them. Enzymes that can break down organic molecules like carbohydrates, big polysaccharides, can break down proteins, can break down lipids. And so here we have the lysosome merging with the recently phagocytized food. There goes the enzymes in. So these two vesicles meet like two bubbles. Can you imagine two bubbles coming together and all of a sudden, boop, one bubble forms? Yeah, that's kind of what happens. And so here's the use of those enzymes to start to break down that food, if you will, and those amino acids that are broken down from the protein, those monosaccharides that are broken down from breaking down the polysaccharide, Those fatty acids and glycerol that form when you break down the fat are then utilized by the cell, maybe to make more cell membrane. Because remember, it can't be doing this endocytosis forever. It's got to replace that cell membrane that's been used, for example, right? Otherwise, the cell would shrink, 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 and maybe die. So you got to add more proteins, maybe to the cell membrane. Maybe you got to make more phospholipids in the bilayer, et cetera, et cetera. And then those undigestible waste products, if you will, are released out into the environment, maybe out into the bloodstream where they're taken and released via the urine or some other mechanism that we get rid of the unusable residue. I'm not sure if that answered your question. It kind of did a little bit, I think. You have to kind of think of the cell.
Yes, thank you so much.
Okay, the cell is, it's a microscopic little thing, right? But there's a lot going on. It's a complicated little ecosystem. All those organelles have specific functions and they work together. I'm not saying consciously, but there is an orchestrated workflow going on within a cell. It's quite astounding when you study it a lot and take cell bio and other courses in this field. You just come away with some totally different appreciation of all of the intricate biochemistry and things going on. Wow, it's just mind-boggling. And we are just scratching the surface on a lot of this stuff in A&P. I know I'm throwing a ton at you and your mind is kind of like exploding, but I hope it makes you more inquisitive and you ask questions and that's really wonderful because then you're starting to think a little bit about stuff that you maybe didn't think about before. That's really what college is all about, right? This specifically addresses one example of exocytosis, and this is in relation to what are called neurotransmitters. Maybe you've heard of these chemicals. Sometimes in psychology, you study acetylcholine or norepinephrine or noradrenaline, and that kind of thing. Those neurotransmitters, which are really critical for nerve function, they are manufactured in our cells. And this simply talks a little bit about the role of the Golgi body to manufacture those special neurotransmitters. And this is the release of the neurotransmitter out of the cell. So it's not always residue waste products that are being released by cells like we saw in the preceding slide here. These could be directly usable molecules that the body needs. And in this case, for like neuron function, for nerve impulse conduction. Cells have to make those neurotransmitters. Where do you think enzymes come from that help break down organic compounds? Well, your cells make those enzymes. They don't just come out of the blue. And to make them is one thing, but then you got to release them out into the circulation perhaps, or out into the lumen of the stomach to break down the protein or whatever it might be. And so that's what many cells do. They are releasing substances via exocytosis, making them internally and then exporting them outside the cell. Transcytosis, I know you mentioned that a moment ago. I just asked you to kind of look this over a little bit. I didn't lecture much on it. I'm not going to spend time lecturing on this right now, so you can read it over if you want. How about other questions? This chapter covers lots of different stuff, doesn't it? Goes into the cell cycle, talks about control of cell division. It is a potpourri of different topics. This chapter covers lots of stuff. Anything you'd like to review in chapter 3? How far have you gotten in chapter 3?
I did all of it, and I just wanted to say thank you for posting your lectures in Blackboard, because I have an hour commute to and from JCC, so I've been listening to them in the car, and that's been really helpful. And then if anyone is struggling with the cell cycle and remembering, I found a little like monomer on YouTube, and they were saying it's I prefer milk and tea, and it helps you remember the phases. So, the interphase, prophase, metaphase, anaphase, telophase. So that's helpful if any of you guys are struggling with that.
Yeah, that's great, Paige. Anytime you come across, YouTube videos or mnemonics in any way that you find helpful, and there are lots of them out there, yeah, let's share those. And the other thing that I was just going to mention when you were talking about that, this semester in my microbiology class, there's a student who I had I think I had her last spring. And what she did last spring and what she's doing now in microbiology is she's got a Facebook page that she's inviting people to come and basically do like study groups. So this would be really a nice thing to do if somebody wanted to, within our small class, have a study group within Facebook or whatever platform you want to use. Now, of course, it does require somebody to sort of take the lead but I was just thinking, if you did have that sort of Facebook study group type thing, then Paige could share stuff like that with all of you, and you could bring things in that maybe you discovered that would help you or help others rather with the material. So it can be more enjoyable because you work together, you ask questions, you learn from one another. That's why I just think study groups are really great. It's hard now with COVID. You can't like meet in the study room on Wednesday afternoon like you used to be able to do, but it can just be a really fun and enjoyable experience if you get a study group going. Yeah, so I feel sorry for you listening to me on your way to work or school in the morning and then going home again probably, but I think what you're finding, Paige, probably, you tell me if this is true. You can't obviously study when you're listening or driving, but just hearing it does plant the seed. And then when you sit down at home and watch the lecture, okay, it's kind of not maybe review, but it is a little bit of review because you've heard of it, but now you're actually watching it or you're referring to the diagram or what have you. So yeah, that's really an interesting idea. And you're making use of your time. Otherwise, you're just gonna imagine not listening to the radio or whatever. This way you're learning so much. Yeah.
Is there anything that you really think we should touch on as we study for next week with the and how do I say this, integumentary system and the skeletal system?
Okay. What do you mean
by?
by?
Oh no, that's too far. That's too far.
Yeah. I was going to say, Paige, you're a month ahead. Man, you are good.
I know. I'm a mess. Okay, I guess as we look forward to tissues, our cellular metabolism.
Well, I'll tell you what. Don't get ahead of yourself too much. Yeah, the cellular metabolism chapter 4. I'm gonna just tell you straight up. This is a little harder chapter because there's a lot of biochemistry in this chapter, and it often causes lots of consternation for students. And again, I'm not trying to scare you. I'm not trying to freak you out or anything, but you just need to be aware that chapter 4 is going to be one of those chapters that you're going to have to kind of ask questions and go over again and again. So to answer your question, follow the PowerPoints, or the Zoom lectures, I should say, and the PowerPoints. PowerPoint slides for chapter 4. But seriously, you've got to come and ask questions. If something's not making sense, I cannot read your mind. I mean, I'm trying, but I have a hard time. You've got to tell me, hey, this is not making sense to me. Can you go over blah, 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 or whatever the case may be. But chapter 4, I don't know what to tell you, what to focus on. Let the Zoom lecture guide you. There's all this stuff is pretty important stuff. I would say, though, when you get into glycolysis and the citric acid cycle and active transport, which a lot of you have no idea what I'm saying because you haven't gotten to chapter 4 yet, but these are complex biochemical pathways. Don't get all caught up in the names of all of the chemicals. Okay? That's, I guess, one thing I would say. Do not get all caught up in the names of the chemicals. That might help you. Yeah, you've got to go in the content section of the course shell. And there's a folder that has the Zoom lectures. And there's another folder that has the PowerPoint that are non-narrated. By now, you should be all familiar with the course shell and what's in it and where to go to look for stuff. I mean, don't be afraid to ask, obviously, but make sure you're using all of these tools in your toolbox, right? You know, we can use some of this time to talk about chapter 2 as well, right? The chemistry chapter, because a week from today, you have your chemistry quiz. So I don't want you to forget about that either. Yeah, Jess, I think.
Okay, if you're open to, oh, I'm sorry, go ahead.
No, I was just going to say, Jess made a comment in the chat that somebody had kind of a weird microphone noise coming. I heard that too, but I think that's gone now. Go ahead, Paige.
If you're open to talking about the chemistry section, I'm just looking at this, test study guide that was on Blackboard. And the one question is name and describe 4 basic types of chemical reactions. If you could touch base on that. It's been a while since I've been in chemistry.
Sure. Okay, I'm pulling up the PowerPoint. And so what that objective specifically was referring to, yeah, I guess I don't have a slide of that, but I can tell you exactly what that refers to. In chapter 2, on page, where is it? 68.
Is it 68?
Yeah, under chemical reactions. Yeah, there really isn't a slide on this, but what they talk about are synthesis, decomposition, exchange, and reversible reactions. So if you have your book, flip to page 68. The heading of the section is chemical reactions. And it's simply saying that if you take two reactants, we'll call one A and one B, and you combine them together, as shown in the top column, right-hand column of 68, synthesis means to build, to produce, to manufacture something. So the union of those two reactants forms a product called AB. This could be as simple as glucose bonding to fructose to make the disaccharide sucrose, sugar. Two monosaccharides combining to make a disaccharide, right? It could be three fatty acids combining with the glycerol to make a triglyceride or fat. That's a synthesis reaction, right? Decomposition is just the opposite. It's taking AB and degrading it into its A plus B reactant components. It's like taking sucrose, breaking it down into the two amino acids, glucose and fructose. Or taking the protein and breaking it down into its amino acid building block molecules. Reversible reactions are just where molecules sort of exchange positions to form unique different molecules. So again, AB plus CD yields AD plus CB. One example I can give you of that, I don't know if I can write this down and show you on the screen here, hang on. Let's see here. Okay, here's an example. Well, can you read that? So NaOH, sodium hydroxide, plus HCl, hydrochloric acid, yields salt water. That would be an exchange reaction. NaOH plus HCl yields NaCl plus H2O. You're just rearranging the molecules, right? To make different molecules.
Okay. So for our class purposes, we aren't going to need to know like what is causing these bonds to break apart or anything, just to be able to define like the difference between these, right?
Yeah, just know generally what the major differences are. That's all. No, you don't need to know anything about why or how bonds.
Okay, that's the main thing I was.
No. That's chemistry. That's chemistry. Take college chem. Yeah. And the reversible reaction is just what it says. You can take reactants to make products, or the products can degrade back in the reverse order to make the reactants back together again. It's a two-way street, if you will. How about other questions on chemistry chapter? I think we talked a little bit about this maybe last Wednesday. I can't remember when. Don't be afraid to ask questions. John, you're not coming in clearly for some reason. Maybe that's the chipmunk, Jess, we're hearing, huh? I don't know. Okay, go ahead, John. Were you going to say something? 1 minute. Okay, he's rebooting. Yeah, I didn't know if John was breathing in some helium or what was going on there.
I don't mean to like hog the class, but I guess nobody else is asking a lot, so I guess I don't mind.
No, don't.
So the one I'm having a hard time finding this in the chapter, maybe I'm just like skimming too quickly. But one of the questions was, what is the difference between an atom's molecular formula and its structural formula?
Yeah. We talked about that last week, but we can talk about it again.
Okay. Is that just like the weight and the mass and stuff like that?
No. If the question is, what's the difference between an atom's molecular weight and an atom's atomic number, then that's one question. But I think you asked, what's the difference between a molecule's structural formula and its molecular formula? Those are two different questions. So for example, let's go back to the PowerPoint. Okay, I'm gonna find, okay, so on page 66, there at the bottom of the right-hand column, they talk about structural formulas where they're depicting single lines between elements like the two hydrogens. This is a single covalent bond. This is, of course, a double covalent bond. So this is referring to the structural formulas. Here's the molecular formula. This is what you're more commonly acquainted with regard to water, for example. Everybody on Earth knows H2O is water, I would hope.
Okay, and how would we sort of word the difference in an exam? Like, would we, just say that one shows like the makeup of it and one is just like an abbreviation? How would we differentiate those on paper?
Yeah, the chemical formula of the molecule would be a listing of the elements. And by that, I mean the numbers also, the different elements and how many there are. So for example, CO2, 2 oxygens and a carbon make up that molecule. In the case of water, 2 hydrogens and an oxygen make up that molecule. Glucose, the chemical formula of glucose is C6H12O6. That tells me how many carbons, hydrogens, and oxygens are there. What it doesn't necessarily tell me, however, is the structural formula, which is an indication of the types of bonds, i.e. single and or double covalent bonds within the molecule. So one just tells me the number of elements in the molecule, what elements are there and what number are there. And that's the chemical formula. That's what we're most familiar with. And the structural formula would be something that chemists would be more interested maybe in looking at in terms of how those electrons are being shared between the elements. Rarely do we in A&P, for example, talk much about the structural formula. Once in a while, I shouldn't say never, we do once in a while. Chapter 3 and chapter 2 do. But most of the time, we are more interested in the chemical formula. In other words, H2O, CO2, C6H12O6, whatever the case may be. Now, you can get even more complicated if you want, and you can talk about three-dimensional space-filling models, which is somewhat similar to what you see here, only now we're using these two white spheres that connect to an oxygen that we can sort of make up a three-dimensional model of the molecule, which again can be helpful in understanding its properties. But like a double helix, you've all seen a double helix model, right? Double helix. Well, that's a space filling model that maybe was in your high school bio class or lab. It's a physical model that somebody's constructed to give us an idea of the shape. Three-dimensional shape. Okay, I don't want to beat a dead horse here. So, other chemistry questions? John, you're back again. You rebooted. All right, you have any questions?
I switched to the laptop here. So 2.6 is what I was saying is going to show up on the exam, correct?
2.6, what is that?
Organic substances common in the body.
Oh, is it, you're going from the objective list?
No. I'm still in the book.
I don't know. I'm confused. What 2.6? Are you talking about section 2.6? What is 2.6?
Oh, it's table 2.6.
Table. Ah, gotcha. Okay, that table on page 71 is not something you need to memorize.
Okay.
No. I think it's helpful to understand that there are a lot of interesting inorganic ions that have a variety of roles to play in our physiology. We will be talking more about some of these. By inorganic, what does that mean? Anybody know? Inorganic? What does inorganic mean? Well, what does organic mean? What are the organic molecules? There's 4 major groups. You studied those in chapter 3.
1, 2, 3,
4. Or maybe chapter 2. Guess chapter 2 has them.
4, 7, 8.
Carbohydrates, lipids, proteins, nucleic acids. Those are the 4 major organic molecules. What do they all share in common that makes them organic? The presence of carbon and hydrogen. Lots of carbon. Inorganic means without carbon. So as you peruse through that listing of ions, which are charged molecules, there is not enough carbon and hydrogen there for them to be considered organic. So things like hydrogen ions, H with a superscript plus. We talked about the fact that if you have lots of hydrogen ions, that could be indicative of an acid being present, right? So hydrogen ions play a super important role in pH in the body. You'll be learning about calcium ions a lot in this course. We will be talking about how calcium ions are stored in bone, how calcium ions are important in blood clot formation, how calcium ions are critical for muscle contraction. There are a whole host of physiologic principles that rely on adequate calcium ions being present. And so as you kind of encounter those ions in the next 14 weeks or so, you're going to get more comfortable, more familiar with these. This is just a nice table you can come to if you ever want to and look up the major common inorganic ions in the body. And they do a nice job kind of listing what their importance is. But do not worry about memorizing that table.
Okay.
Yeah. So the study guide that Paige was talking about,
Right.
are you talking, Paige, you're talking about the objectives,
The
chapter
objective grade.
objectives?
Yeah, I have the study. This has been really helpful. It's the A&P study objectives, and it just says in each chapter like what you should be able to answer by the end.
And so Alexis is asking, where did you get that?
It's right in a Blackboard shell under in your Blackboard shell. So
Honestly,
to bring the class.
And everything you need in here is under content, pretty much like everything you need for this class.
Content and things you need to bring to class.
Yeah, so let's go back and let's look at the okay, I've got to hang on 1 minute. I'm going to get the Blackboard course shell back up.
Paige, did you read all of chapter 4?
This is where you guys by now need to know where to go to get what
Yes.
in the course shell. We're in the second week of classes. You got to be looking here and knowing what's what. So this is the course shell. I'm going to go to the student view. So this is what you're going to see when you go to the course shell, right? This is what you see. Everything other than the Zoom link to the Zoom room, which obviously you've all found because you're all here, go to content. Everything's in content. Here's the course syllabus. Here's the master course syllabus. Here's the Zoom video recordings. So after today, I'm going to record. I am recording this Wednesday class. I will upload it to YouTube. I will save it to my channel, and then I will link it to this in this folder. And I will say February 3rd, 2021. So that's what you'll see. In addition to those Zoom lecture recordings, also here are the various Zoom lectures. You should have already looked at 1. You should be finishing up 3, and I just freed up 4 while we were in class today, which is on the docket for next week and the following week, according to our syllabus. I will free up chapter 5 and 6 and 7 and 8 and 9 and all the rest as we proceed through the semester. I can free them all up now if you want them now, but I'm thinking to myself, you're not going to get into chapter 14 until the 1st of April. So you tell me, you want me to free them up, week by week, or do you want me to free them up all at once? I don't care. I'm just trying to make it kind of logical. Maybe I should free up like the next 2 chapters. So if anybody wants to look ahead, they can do that. I'll try to do that. Okay, so again, these are the Zoom lecture recordings that I did last semester. And you should be looking at those, watching those, taking notes from those. Let's see, what else was I going to show you? Didn't mean to stop sharing the screen here. Let's go back to content. And then here's the study objectives, which I have for all of the chapters. These are just very general overview objectives to help provide a fundamental guide through the chapter. They're very general objectives. So now Paige, when you were talking about the study guide, and also John, you're talking about this folder perhaps? Yes?
I wasn't.
You weren't.
I have both of them though. However, I do have both of them. The study objective by chapter and the one you are now.
Well, Paige, what were you using?
I have only printed out the study objectives so far.
These?
Yes, yes. Study objectives by chapter.
Well,
Yeah.
no, yeah, but you said, I thought you said like study guide earlier.
Well, I just used that as a general term. I mean,
Okay. Yeah, you have to each determine individually, how you use the stuff. I provided to you. I think that these study objectives are good. I think they can provide a fundamental framework for preparation for quizzes and exams.
So I don't mean to cut you off, but can you use, like I asked you for chapter 1, in which I did, just answer these just answer them as questions, like what is matter, what are elements, just answer the question, correct?
Right. Yeah, just answer the question, right? You're developing your own study packet, right? But I'm saying, while this is really an excellent overview, when I say down here, what are carbohydrates? How do they function in the body? Be familiar with the different classes. This involves a lot of digging, and you've got to go into the chapter. But yeah, if you answer those, that's a wonderful way to prepare for the chemistry exam. Absolutely.
Okay.
When I say understand the pH scale, that's a pretty broad, sentence, right? You got to get in there and you got to look at it and say, oh, it goes from 0 to 14. 7 is neutral, right? As I go from 7 down to 0, that becomes more and more and more and more and more acidic. So I go from 7 up to 14, it becomes more and more alkaline. So, yeah, I mean, the goal for me in making these objectives up was to give you some guidance and allow you to kind of develop a study packet. But again, these are general objectives, but they're good. Another thing that I think I mentioned to you guys was, let's see, the student study outlines and answers.
Yeah.
So if we, for example, look at chapter 2, chemistry chapter, notice there's just a chapter 2, and then there's a chapter 2 answers. Each chapter has 2 files, one without the answers, one with the answers. So if we go to the one without the answers, they're all Word documents. These are in some ways similar to the study objectives we just talked about, only this is much more in depth, more detailed. This is asking you to list the 6 examples of bulk elements. This is asking you to define what kind of charge an electron carries. This is telling you to determine or define what the weight of an atom is, right? Due to the number of protons and neutrons. Isotopes are what? So yeah, this takes you through each section here and gets into the nitty-gritty. If you can answer these, excellent, because you can check your answers by going to the answer of chapter 2, right? Another document, and there you will indeed find all of the answers to those blanks, right? The 6 examples of bulk elements are electrons carry what kind of charge? Negative. Yeah. So I'm not saying you have to do these or you should necessarily do them, but you know, if you think that would be helpful to you, then by all means.
Mhm.
You just have to develop your own study mechanism, I guess. Strategy. Any questions on chapter 2?
I'm done.
Anything on chapter 3? Some of this stuff is and should be reviewed. You know, the stages of the cell cycle. You, I'm sure, learned that in high school. So it's not brand new. Shouldn't be brand new. So as you prepare for that chemistry quiz, work through the objectives, for example. That generally will provide you in, you know, with enough to do well on the chemistry quiz. Okay, well, I will be in the Zoom room till 2:30. You guys can hang around. We can talk one-on-one or whatever. If not, I will see you guys, I guess, next in review Tuesday, some of you, right?